This is question 8 of the core component in-class question book. And we're on to the second question of enzymes. The scientists Devulapalle, whoever that was, and Mooser, that's a better name, carried out experiments using the enzyme glucosyl transferase. Never heard of it. But it's got A's at the end, so it's definitely an enzyme. And it tells us what it does anyway. Catalyzes the polymerization of glucose molecules from dietary sucrose. So it makes a polymer of glucose. So what other polymers of glucose do we know? Sort of, glycogen, starch, and things like that. And from dietary sucrose, dietary that you eat. These polymers of glucose form part of the plaque on teeth that can lead to dental disease. Okay. Name 2 theories that can explain how glucosyl transferase can combine with its substrate sucrose. So this is AO1 stuff. Can you remember the names of the 2 theories that we talk about when we talk about enzymes? Yes, you can. Lock and key and induced fit. So you need both of those for 1 mark. That's straightforward. Explain why the rate of a chemical reaction is increased by the enzyme acting as a biological catalyst. Again, straightforward. AO1. I know about enzymes. I know that they lower the activation energy. Lower the activation energy. So good so far. Here's a data table. They showed that the presence of iron 2 sulfate, whatever that is, affected the activity of the glucosyl transferase enzyme, and the results are. So this stuff affects the enzyme activity. So that sounds a bit like an inhibitor. And as we increase the concentration of the stuff, the activity in totally incomprehensible units goes down. So if we drew a graph of this, so when you see a data table, it's always good to kind of draw a little graph in your mind of the kind of trend in the table. So as we increase the iron to whatever concentration, then the activity, so it starts off at 60, goes down pretty quickly, that sort of shape. Okay. They concluded that iron 2 sulfate was acting as an enzyme inhibitor. Further investigations showed that iron 2 sulfate was reduced. The effect was reduced at higher concentrations of sucrose. Right, okay. That's a little gem of information. The effect of the inhibitor was reduced at higher concentrations of sucrose, which is our substrate. So, more substrate, less effective an inhibitor. That should ring with one of your 2 types of enzyme inhibition. And then it just says explain how it could inhibit glucosyl transferase. So this is sort of AO2 stuff. They're giving you some data to look at, and then you're relating your knowledge to this data. We've got 2 types of inhibition that we're thinking about, competitive and non-competitive. And the information that's going to decide between the 2 is, as we increase the concentration of the substrate, this is affecting the inhibition. So we're going to go for competitive inhibition. Okay, so we're actually going to get a mark, one of our 4 marks for saying competitive inhibition. And then it says explain how this the iron sulfate could inhibit the enzyme. So competitive inhibitors, if we just trot out our knowledge of competitive inhibitors, so the iron 2 sulfate, it could have a similar shape to the substrate, or alternatively we could say complementary shape to the enzyme, or a complementary shape to the enzyme active site. We can't say same shape because other parts of the molecule might be slightly different, or we can't say same shape to the active site because it's complementary, so fitting in, that sort of idea. And what happens, it fits into the active site, or binds to the active site, so fits into the active site and prevents the entry of the substrate. Prevents entry of the substrate, which in this case was sucrose. So the idea of blocking the active site from the substrate. So we said 4 things, and we've actually got a line spare still. Then over the page, AO3 stuff, which is making suggestions of uses of things. So suggest a possible use of the findings. Well, we need to go right back to our start here. If you can't think of anything, go back to the start. And then why are we looking at this? Polymers of glucose form part of the plaque. So if we use this iron 2 sulfate, it stops as much plaque being formed. So let's use this in toothpaste or something like that, or in the dental industry, something along those lines. So I'm going to put use iron 2 sulfate in toothpaste. It doesn't say whether it's toxic or anything like that, but we don't care about that. It's just a sensible suggestion. Put it in toothpaste, mouthwash, sugary drinks, and so it stops as much plaque being formed for 1 mark.